molecular biology? What do you think? You take the class? What do you think is molecular biology?
Biology taken down to the molecular level.
Excellent. That's exactly what the molecular biology is. So basically, it's the study of life at the molecular level, right? So then the next question is, what is life? Well, this is a big question because we're going to study we're going to study life at the molecular level. But what is life? Well, I need 4 volunteers, okay? I have a beaker here and some chemicals. So, okay, I need 4 volunteers, maybe 2 male students and 2 female students. Who wants to help me for the first class? Thank you. So we have 1, 2, 3, and we need another male student. I'll do it. Excellent. Thank you so much. So basically, yeah, come here. Come over here. And so we are going to divide it into 2 groups. Okay, so when one group is doing the experiment, the other group is going to be the witness. Just to see whether they are doing things right, all right? And I want you to watch. Okay, so you want to maybe you two a group?
Okay.
And while they're watching, so you start first. Tell the class what you have here.
Some baking soda.
Some baking soda. Okay, pour some baking soda in the beaker. Okay, that's enough. Okay, so what is this?
Vinegar.
Vinegar. Okay, thank you. So can you pour some vinegar into the beaker? Watch. You can pour more. Yeah, that's it. That's good. Thank you. What did you see? You see some bubbles, right? So now your turn. I only have 1 because so you can come over. Thank you.
Just do the same thing.
Yeah, you can do the same thing. So now you can let's just make it bubble out. I'm a chemist. Sorry. But I'm glad that I see some of you are double major in chemistry and some biochemistry. So yeah, why don't you pour some here? Oh, yeah. What did you see? Very excited. Do you want to try too?
Sure.
Oh, she poured a lot of baking soda in, so it's difficult to make it. Let's add some more vinegar.
Okay.
Okay. Do it slowly. Wow, that's interesting, right? Good, thank you guys. So, what did you see? So basically, they told us that we had the baking soda here, in case you cannot see it, and we have white vinegar here. This basically is the chemical components of the baking soda, right? And this is the most important thing in vinegar, which is an acid, and here is a kind of a base. When they put them together, what do we have? We have this salt and we have a bubbling, right? So this basically is a carbon dioxide, and now this is soda, right? We can even drink it. So there's 4 of them, we have 4 brave volunteers came here, and each time when we have this baking soda and the vinegar together, they interact with each other, right? And the proof is you saw the bubbles, right? The bubbles were carbon dioxide. Is it possible that I pour the the baking soda, and here we have vinegar, still some vinegar left there, I think. Can I say, don't do that, okay, I pour the I pour the baking soda, and can I say, baking soda, listen to me, don't make any bubbles. Can I do that? I cannot, right? I cannot. But when I ask the students who come here, right, they can refuse me, right? Actually, most of you did. I asked you to volunteer. Most of you did not volunteer, right? But, we have 4 came here, so that's good. So you can, when I ask you to come, you can come, or you can decide not to come, right? However, if I put the baking soda and the vinegar together, they cannot say to each other, say, no, I don't want to interact with you. I have also here sodium and chloride. So to interact, they form sodium chloride. And I don't want to try this reaction here where I have a fire, right? Anytime you can command, you go home, take your table salt out before you cook them, and you say, sodium, I command you to separate from chloride. Can you do that? Or can someday chloride says that, sodium, I'm so tired of you. Get out of here so I can be alone. Let's separate. Can they do that? So what is life, right? Chemicals, we saw that actually they have to, they have to, if their chemicals, their properties, tell them or not tell them, actually don't tell them, depending on their own characteristics, they will interact with some chemicals, and once those, the ones they supposed to interact will always interact, right? They cannot choose to say, I want to interact today, but not tomorrow. Or I want to stay together today, but not tomorrow. But the life is different. Life is different, even simple lives. What is life really, right? When we think about life, we may think about self-replication and metabolism, growth, and even death, right? We can have other things. And, when we see something alive, we can tell, right? We can tell the differences between something alive, like, each of you from the chairs or tables, or even, these vinegars and the baking soda, right? When we see something alive, we can tell, it's alive, it's a living thing. Even when something is dead, right, it's not moving, we can still tell that at least once it's alive, right? So what is really making something alive versus dead versus non-living, right? How can we tell? So when we talk about molecular biology, yes, we're going to study life at the molecular level, and we are going to look at the hardware, so-called. So they are basically the molecules, exist in life forms, including big molecules like DNA, RNA, proteins, and there are also small molecules, lipids, water, and ions. So we're mostly going to focus on the big molecules, DNAs, RNAs, and proteins, right? When we look at the DNA, we all know that DNA has a double helix structure. This is, we all know that, very familiar, right? But when we have a closer look of a DNA structure, so here we have the sugar phosphate backbones, right? Sugar phosphate backbones. And we have a bases in between. We know that what is the most important for DNA are actually the bases, right? The bases are very important. The different composition and the sequence of the bases are what determine the function of DNA. However, look at this. In between these base pairs, right? So we have a double helix, right? When we know the basis of one strand, by base complementation, we can know the sequence of bases on the complementary strand, right? However, in between these base pairs, look at this here. What do you see? I'm sorry? What did you see? Hydrogen bonds is between these base pairs. So between the base of one strand and the base of the opposite strand, right? I'm asking here in between here. So one pair here, another pair here, this pair and this pair. What's in between here? The directions, do you see anything here? Any chemical bonds here?
Empty space.
Empty space. It's strange, so basically here, what is important, because it is very important, what is this after this base pair, what is supposed to be this, right? Well, actually, we don't know, because here, this is empty space. Basically, this is telling you that although it's important that a specific base will follow one specific base, right? However, there's no chemical bonds. No chemical bonds, or even like weak chemical bonds, like hydrogen bonds, even those hydrogen bonds, you don't see that. So this is an empty space. There's no force, no chemical forces, no physical forces can tell the organism that, which base will, for example, which base will follow this base? You would have no idea. Which base will follow this place, right? We don't know because, not like sodium and the chloride, when they put together, they have a chemical affinity, they will interact with each other. But here, the sequence of the bases, so what base will follow a base in front of it is not determined by chemical bonds, right? Here's the empty space. So what determines the specific basis a gene would have? What is the decision factor there? Well, that's DNA, right? When we look at the RNA, so here's the DNA if we look at the one strand, and if we look at the RNA, the same thing occurs. So there are no chemical bonds between the bases. What links all the bases together is the phosphor sugar backbone, right? But the sugar backbone, is the same for each place. They are the same. There are no changes in the backbones, but the bases keep on changing, right? So what is determining the sequence of DNA? And when we look at the protein, we see the same thing. What is important between the proteins? The proteins are linked by peptide bonds, right? But the peptide bonds are all the same. What makes one amino acid differ from another is the side chains. So side chain here is using R1, R2, R3s to show. And there are actually no chemical bonds in between those side chains. So what determines the sequence of amino acids in a protein? Right? We know those are very important, but who tells an amino acid that, now it's your turn, go there, right? Follow somebody there. So for each base, A or T or G or C, any other bases can follow. And for each amino acid, any of the 20 or 21 or 22 amino acids can follow. There are no chemical or physical bonds to determine the sequence of DNA or RNA or protein, although we know that the sequences are essential for the functions of the gene. Right, so this actually brought us to the, the other property of life. So-called the software of life or information of life. This is like language, right? So language, we have words, sentences, paragraphs, and of course we have grammars. For DNA, actually people even use the computer program that used to study language. They go to use those programs to analyze DNA. And they found that it's strange. So with DNA, if a DNA will code for protein, there's a coding region and there are non-coding regions, right? And there are actually a lot of DNA segments in our genome, like a human genome, right? Actually about 99% of our human genome does not code for any protein. But when they use the program to analyze language, they find that actually the non-coding region of DNA is much, much more similar to languages. which is very surprising, right? So here I have, I love you. I'm not lying, I do love you. Where this and this is the same in the same styles, they contain the same letters, right? But this makes sense, right? But this is gibbish, right? The only difference is that the order or the sequence of the letters are different. So depending on the order, the same letter can make sense or not. Well, the same is true for DNA, RNA, proteins, right? We can have, this is just the codon table, right? We can have the same letters, but the meaning can be totally different, even opposite, right? So here, AUG is a starting codon, so to tell a gene to start translation. And different order of these three letters, UGA and UAG, actually they code for stop codons. So that's the codon to tell the cells that that's the end of translation, right? So when we look at the genes or genomes, actually now we have even the human genomes have been sequenced. So when we look at the genome or the DNA sequence in more detail, so we find that there are informations actually encoded in our genome. Some of the information is sequence dependent, like the codon, like AUG is a starting codon and UAG would be the stop codon, right? There are also information is sequence independent. So that sometimes is descent dependent, sometimes is structure dependent, I'm sorry. So we have in our genome, actually there are multiple layers of information, not only the primary sequence, but also how how the DNAs, RNAs, or proteins, how they are folded together, right, to form 3D structures, and sometimes even 4Ds, changing with time, right? So what is life? Really, what is life? Back to the question, right? Is life DNA? Is life RNA? Is life proteins? Or, well, the sum of them, right? DNA plus RNA plus protein. Is life mere chemistry and physics? What can we learn from the chemistries of DNA and RNA and the protein about the nature of life and maybe the different relationship of different life forms and even the origin of life? Okay, these are the questions that we will touch upon, and I hope that what we learned in the class will really make you to think about life, right? What is life? Because we are studying life at the molecular level, right? This is a picture I took from YouTube. So this is an exercise. The title says, one adult and two kids were killed in this accident. What happened? Their DNA has not been changed. their RNAs are still there, and their proteins are also there. You know, the moment they were hit by the car, I don't know how many minutes it took for them to die, or seconds for them to die, right? All the chemicals, all the DNAs, all the RNAs, and all the proteins are still there. You know, 1 minute, they're alive. The mom, adult, I don't know, they didn't say mom or dad, so I don't know. So at least one is older and the other two are younger, right? Supposedly they're all healthy on the car. What happens? You know, we have all the molecules still there. Well, they are somehow, you know, the organization of those chemicals are changed. So life no more, right? What is life? So this makes us, I hope that, that at least makes us think whether life is just a chemistry and the physics, or there are more than that. Is, life just a DNA, RNA, and proteins, and the sum of them, or there are more than that. Analogy is like we can think about storybooks, right? Is a storybook the same as a story inside or not? Oh, we all use phones now, right? So is the phone and the phone conversation, or what you say to the phone, the same? What is the relationship between the storybook and the story, your phone versus your conversation, our genes, our DNA, proteins, and life? Molecular biology, back to molecular biology. We study life at the molecular level, right? That is genes and their activities. And we are going to look at the DNA and the proteins and the making of DNA, that is the DNA replication and transcription. I heard somebody say transcription, right? Wants to know about transcription. So we have a transcription and processing of RNA, and then we're going to talk about translation. We're also going to emphasize things about the techniques, and actually that is part of the reason we have a paper presentations, right? So the central dogma says that the information will flow from DNA to RNA to protein. And here is a diagram. So saying not only DNA can be transcribed into RNA, RNA can be made into proteins. We also heard that the DNA will self-replicate, right? We'll see if DNA indeed can self-replicate. So we'll see a lot of details on those things. And so the courses were going to be organized by 2 lectures by me and 1 paper presentation per week. As I said earlier, the first paper presentation will be done by me as an example. You have to be here, okay? The lectures are your most specific guide and is very important. I sat in a How Brain Works just 2 weeks ago. Actually, this is the last 2 weeks. I heard that the optimized way of learning is coming to class, because this is like you have multiple things included. you're listening, you're seeing, and you have classmates. So that's the optimal environment for learning. So it's important for you to be here. Right? What is very important in the class is I hope you to think. And whenever you have questions, okay, even when I'm talking in the middle of my sentence, ask questions. If you have questions, raise your hand. Gradings. So we have 20 points for participation. And so to be here is very important. It's a very important part of your grading. Okay. Each time you miss the class without being excused, 1 point will be deducted. And for paper presentation, that's another 20 points. We have quizzes, 20 points, quizzes, clicker quizzes. So you need to have a clicker. If you don't have a clicker yet, you can go to the bookstore, either buy a clicker or to purchase a subscription. Then you can vote with your cell phones or your laptops. Although honestly, I don't like to have a laptop in the lab, in the class, because some people will surf, they cannot control themselves. Okay, then we have midterm and final, each 20 points. I have also a bonus, so you can write essays, and each week you are allowed to write one essay about what is taught in the class, or you can write on small things or big things. Big things meaning what is life and where life came from, blah, those kind of things, right? We're getting out of time. So the gradings, the participation, right? Participation. It's important for you to raise questions, to be here, answer questions. For presentation. So what is important really is to be here, ask questions, to be active, because at the end, right, if your grade is maybe if it is between an A and an A minus, and I find that you are very active in the class, I will boost you up to A, okay? So it is important to be active. For this Friday, yes. Bring your clicker Friday so we can test the clicker, okay? So the course is recorded. The course is recorded in integrity, and you can listen later on if you want. And I'm also going to post my PowerPoint presentation on the web after the class. Okay, see you guys Friday.